Hi all, Tony here from Immortality, IMT cryptocurrency for the longevity field. Today we want to give a presentation what is believed to be a major breakthrough in human radical lifespan extension, and we want to present that to you today with the whole backstory on how we got there. Nature has provided examples of immortality. Certain animals exhibit biological immortality. There is no example in nature of pharmacological immortality. There is no animal in all of nature, in all the species of nature, that has ever lived in the history of Earth that go around looking for a magic molecule to obtain immortality. Pharmacology is not evolutionarily proven, which is a big question mark over pharmacological strategies. Secondly, there is no damage theory of longevity in nature. There are no animals that are immortal because they are impervious to damage. None. The only example that nature provides of immortality is by means of regeneration only. Therefore, we're not going to concern ourselves with pharmacological processes to immortality. We're not going to concern ourselves with damage theories. The only way that it's done in nature is through regeneration. It's the only means that nature has devised biological immortality. We at Immortality are in the third category, and that is following nature's way and what nature has done. Now, a lot of people are dismissive of immortality, but however much they want to disagree or they want to act like it's fiction, it exists in nature. It actually exists in our reality. And so why should it be ignored?
Well, I'm Aziz. It's nice to see some of you again. It's been a while. And we've been continuing our work with the so-called immortal worms. And since those first set of videos came out, a lot of people have been in touch asking about how do we know whether these worms are really immortal. So these are planarian worms, flatworms that live in they're free-living worms that live in freshwater lakes and ponds. You can probably go to your local park, and if you have a pond or a river, if you go to the river's edge and the bank, hopefully you'll be able to find some of these worms. The reason we use them in research is because they have amazing powers of regeneration. They have a population of adult stem cells, and this population of stem cells allows them to regenerate. And some people refer to them as immortal. I think the best demonstration of that is to show you the regenerative capacity of one individual worm. So in there, I've got a population of worms, boxes and boxes, that I can show you, that all come from one worm that has been cut again and again into smaller pieces.
But literally cut like?
Yep, we've taken a worm and we've cut it into several pieces. Each piece goes and regenerates a whole new worm, and we can just keep doing that. So that's really the best demonstration that they might be immortal. And this led to a guy in 1814, a guy called Dalliel, referring to them as immortal under the edge of a knife, or almost immortal under the edge of a knife. So in here we have our stock of planarians, flatworms, and we keep our stock here so when people want to do experiments, they can come and take worms, take them to the lab, and do various things with them, chop them up, inject them, knock out genes, look where different genes are expressed and what they do. But the stock that we have, that we work with mainly, came from one individual worm. So I'm just going to show you at the moment how many worms we have from that one individual. You have to promise not to play the Benny Hill music, right? We need to speed it up. They'll start moving soon.
All from one worm?
Yep, all from one worm. Well, they're going to be clones of each other. So during their normal life cycle, the animals actually, when they get to a certain size, fission naturally. All we've done is speed that up for convenience. So every worm in here, because it came from one individual, should be a clone of the original worm. Bear in mind that mutations may have occurred during cell division and DNA replication, so that some of these individuals might have acquired differences from each other. But essentially, yes, they're going to be clones of each other. So it's a shame you can't see any, but often you see them on the side, stretched out, ready to fission. You sort of see them on the edge of the box, and their head's pulling away from the tail.
The first worm, the progenitor, the mother of all these worms. How did you choose that one? Was it a special time, or was it just a very arbitrary? Okay.
It was arbitrary. Yeah, we just picked one worm that we had from a stock sent to us from another group that's been working with these worms for a lot longer than we have, and we just started cutting it. That first worm was probably cut into 3, and those 3 were cut several times, and so on and so forth. So this is one demonstration of how the worms are immortal. We can get all these worms from a single individual. It'd also be possible just to keep one animal happy. It shouldn't die. It should just respond to food, grow a bit, shrink a bit when it doesn't get any food. And as long as it didn't get infected with anything, it should live forever. But of course, we'd like to test that. So as scientists, we've got slightly higher standards of proof. So one way to prove they're immortal would just be to stay here forever and just watch an individual worm and just show that it kept living and keep looking after it. Unfortunately, that's not really practical, at least not for an individual scientist. So there's other ways we can try and test whether these worms are immortal. So if an animal is immortal, there's certain things that it must be able to do. Okay? So for example, with human beings, we get older. So our ability to heal, our ability to replace our skin cells gets worse as we get older. We get crinkly, start to get creases under our eyes, etc. And part of that is because as we get older, our ability to replace our aged cells decreases. And the way we replace our cells, just like the worms, is from pools of stem cells. So we have stem cells just like the worms. But there's a big difference between our stem cells in our bodies and the worms. The stem cells in the worms appear to be able to just keep dividing on and on without any limit, as you can see from the number of worms. All those worms have come from individual worms growing from cuts, making new worms, and they eat and they get bigger, and then you can cut them again and make more worms. So those stem cells keep dividing. Whereas we don't do that. We get fatter and thinner during our lifetime, but generally we get older and older, and eventually, old age is basically a lack of our body's ability to keep repairing the normal damage, the wear and tear that happens during our daily lives.
But humans go on forever, don't they? We do kind of reproduce, and more of us exist, like the worms.
So what that is, that's sexual reproduction. So the way we reproduce is we have egg and sperm come together, and a new embryo develops, and a new individual forms, gives rise to new life. Part of that process is rejuvenation of the stem cells, in a sense. So in a sense, if you like, our germ lines, our sperm and our eggs are in a sense immortal because they keep being used to reproduce, make new animals, and so on and so forth. These worms are asexual. The worms I've shown you today are asexual in the sense that they don't go through that bottleneck of having an individual egg cell and an individual sperm cell come together. They just have a pool of stem cells that can consistently rejuvenate the animal. So what we did is we sat down a few years ago. We made a list of things that we would expect an immortal animal to be able to do in terms of molecules. So we go down and look at the DNA of these animals and how the DNA is maintained. There are a list of criteria that must be true for the stem cells in these animals to keep going to allow them to be immortal. And we kind of put them in order, temporal order, of when they would become a problem. And the first one, in terms of temporal order, is the ability to actually maintain the length, the ends of DNA inside the cell. So in all animals, whenever a cell divides, the DNA needs to be replicated. So if you imagine, if I just take this piece of string as an example, as a model of DNA, and DNA can have this string shape, which is obviously tied into a double helix, and during cell division, we need to make an exact copy of all the DNA in a cell. And this DNA process essentially involves, if I get another piece of string, getting an enzyme called a polymerase and just copying the DNA along. Okay? But there's a problem with that process and the mechanics of the process, which is quite complicated. But essentially what it means is that you need a piece of DNA at the end to act as a primer or a beginning point for this polymerase to bind and synthesize new DNA. And as a result of that process, what actually happens is the very end of each string of piece of DNA, each chromosome, doesn't get replicated properly by this mechanism, and so you lose it. As we get older and our stem cells in our body keep dividing, the DNA at the ends of the chromosomes gets shorter and shorter. The reason it isn't bad news is because all animal chromosomes have protective structures at the end, which consist of sequence that, in a sense, doesn't matter so much because it doesn't code for genes. And these structures are called telomeres, and they're maintained by an enzyme called telomerase. So there's a balance between telomerase activity, which re-extends these chromosome ends, and cell division. So as cell division occurs, these telomeres, these special structures get shorter, and then telomerase comes along and makes them longer again. But there's a limit to the extent to which telomerase does this. And that's very important, actually, for human aging and human health in general, and I'll explain why in a minute. But essentially what happens in stem cells is that you get, the chromosome end gets to a critical length, and the cell decides, well, that's it, I'm not going to divide anymore. Because I've reached this critical length, and if this end gets any shorter, I'm going to start damaging genetic material that is important. It decides, well, the only way to cope with this actually is to stop dividing. And that's because in those cells, telomerase is no longer active to keep re-lengthening the chromosome end. One theory of human aging is that aging is a result of the loss of chromosome ends, resulting in the ability of stem cells to keep, they actually lose the ability to divide. So in cancers, for example, where cells keep dividing, it's almost nearly all cancers, 90% of cancers, have actually reactivated the telomerase enzyme when they shouldn't, and these cells keep dividing. So if you like, aging can be seen as a balance in some respects between aging, okay, you want to live for as long as you can, but you also want to avoid cells that divide out of control. And the way to control that is to put that length limit on. So we sat down and thought about the worms, and if they're immortal, they must have a method for overcoming this problem. So they must have a way of keeping the ends of the chromosomes intact. And if they don't have this, they probably can't be immortal, because the chromosomes ends will get shorter and shorter, and the genetic material eventually would be damaged, and they would not be able to keep going, right? So we hypothesized, this is what you sit down and you think, well, how can we test this? We hypothesized then that these animals would have a method of indefinitely, as in forever, potentially forever, maintaining their chromosome ends. And we went and tested that idea. So the way we do that is we actually went and looked in these animals and looked at two things. One is the activity of the enzyme telomerase, and whether it's there at all, and whether it's able to maintain the chromosome ends. And then we also measured the length of the chromosomes. And what we found was really exciting. We found that if you had a worm and you kept it in culture, but you didn't cut it and you didn't allow it to fission naturally, the chromosome ends just get shorter and shorter. And for us, we take that to mean it's aging, which is obviously bad. But if we then take those worms and we allow them to fission, so we allow them to regenerate, or we cut them, so we artificially cut them and make them regenerate, the chromosome ends actually recover and get longer. So these animals have the ability to rejuvenate their chromosome ends, which is what we'd predict if they were immortal. So one of the criteria they would need to fulfill for being immortal worms has been fulfilled. So we're really happy with that result, because it fits in with our ideas that these animals might potentially be immortal. So the skill which my graduate student, Thomas Tan, developed was to be able to measure two things. One was the actual length of the telomere structure, and to do that he had to essentially take worms, which he knew the age of, or he knew how long it was since they last regenerated, and grind them up, run their DNA, the DNA which corresponded to the telomere ends, on a gel, and take a probe, a radioactive probe, and throw it on that DNA, and that allowed him to measure the length of that end structure. And I can actually show you, if you like, what that looks like. Between my fingers, you can see here one of our animals that hasn't regenerated for about 3 months. And we've taken that animal and we've cut it, let it regenerate, and then measured the telomere length again. Yeah, so when Thomas first came to me and showed me some of these gels, some experiments, I just thought he'd got them the wrong way around. Because obviously they don't fit with what we would necessarily expect of an animal which has a lot of cell division, an animal that's getting older. We'd expect the telomeres to get shorter.
But this is what you expected as well.
Well, yeah, it was very surprising to get such a clear result. So my first reaction to him was, you've just loaded it. You've loaded them the wrong around. So here you've sort of got young and old, you've got old and young essentially. And of course, he had to go back and do all the experiments again and convince me that this is what he was seeing. So that was a eureka moment, yeah, for me. So those first videos were made in 2008, so that's 4 years ago now. And the questions that some of the people asked, like, are they really immortal? Are you sure? Were part of the impetus for doing this work. So obviously when I went to give talks, formal talks to other scientists, but also talk to the public, and also the feedback from the YouTube videos, was that, well, are they really immortal? How do you know? And it was kind of a circular question. And we thought, well, there must be a way, if we sit down and think about it, we can actually get some way towards either proving or disproving that. And this is really the first step that we've made, theoretical step we made towards proving that, the empirical the data that we've collected. So in a way, the viewers kind of help, because we probably would have shied away from this question because it's quite a difficult one. It was pretty hard work to do this work. So we would have shied away from it. But actually, a lot of the feedback we got from sort of public engagement was that, can you prove it? And so we've gone some way towards that. The public drives trends and fashion and pop music and films. So why not science? Science can be a part of that world. I mean, obviously there's the major goal, which is to find out things that are useful, or find out things that advance our knowledge in a way that underpins human health, or underpins medical research, like the work that we do. But I think the public can also be involved in science in a very active way and become interested in science in the same way they're interested in all these other things. Why not? I don't see a problem with that. Because obviously it makes you think that if 20 people write a comment, so are they really immortal, and have a little discussion about how you could prove it, how you couldn't prove it. I mean, however naive some of them are, that really, as a scientist, you have to take note of that and go, well, lots of people seem interested in this. So maybe this is something we should do.
That was Dr. Aziz Abu Baker from Nottingham Science. The ability of perfect resynthesis, indefinite permanent resynthesis, is the only method known to produce an immortal organism. None of the other approaches in longevity, life extension, anti-aging, age reversal can show that proof of concept.
What new information has modern-day biology been able to uncover about planarian stem cells and regeneration? And what do these findings tell us about the human capacity for regeneration? Whether the goal is to regenerate damaged heart or nerve cells, or to regenerate an entire limb, a similar process must occur. A damaged tissue must rewind development, calling on cells that can reverse their differentiation, or rallying reservoirs of stem cells that can generate both differentiated cells and more stem cells. Humans can regenerate certain parts of the body. Wound healing is a poorer cousin of regeneration. But the human liver, for example, can regenerate an entire piece if it has been lost through damage or surgery. Other vertebrates are more proficient than mammals at regeneration. Some lizards and salamanders regenerate lost tails, and salamanders can regenerate entire limbs. However, the organism with one of the most incredible abilities to regenerate is the planarian.
So you can use the salamander to study biological immortality through the regeneration of limbs as a basis. Do you try and make a salamander immortal like the planarian, or do you try and grow a limb on an organism that does not have that regenerative capacity, such as a frog? Can you adapt it? Sure, I hear you say, a planarian can do that, and a salamander can do that, but you'll never achieve that in humans. So here is the breakthrough that we are presenting. Work by Michael Levin and his lab have successfully regenerated an amputated frog's leg. The frog does not have regenerative capacity. The field is already able to regrow limbs in an organism, a vertebrate, that doesn't usually have that capacity.
And so this is just a simple example of what some of these endogenous patterns look like. Here you see, again, a time lapse. This embryo is putting its tadpole's face together. You can see all the dynamic electrical patterns. Here's one frame from that movie. And the amazing thing is, and the reason I love this example so much, is that the encoding is really simple here. The electric, we call this the electric face. The bioelectric pattern of the face actually looks like a face. And this shows up before the genes are turned on to regionalize the face. And so here you can see, here's where the eye is going to be, here are the placodes, here's where the mouth is going to be. All these bioelectrical gradients are a pre-pattern. They're a subtle scaffold that directs downstream gene expression and anatomy. And if we go in and we change these, the distribution of voltages here, you will change the gene expression and you will change the anatomy. That is one way to make those Picasso tadpoles that I showed you about. So so this is a native pattern that is required. It's a normal, natural part of specifying what the tadpole face is going to look like. Here's a pathological pattern. What we've done here is, inject a human oncogene into this tadpole. They will make tumors like this. But before they do, you can already see, using this bioelectric map, you can already see that these cells have a really aberrant electrical potential. They are dissociating from the electrical network. They're basically reverting back to their unicellular state, and they're going to treat the rest of the animal as just external environment. They've unplugged from the collective, and this is metastasis. This is just this is failure to participate in embryogenesis, because the first thing that oncogenes do is uncouple you from the electric network that normally tells every cell what it should be doing. Okay, so the reason we know this is important is that if you then, so you inject tumors like nasty KRAS mutations and so on, if you inject these things and cause tumors, you can prevent the actual tumorigenesis, even though the oncogene is very strongly expressed. So here, this is the same animal, right? So here's the oncogene that we've labeled it with red, very strongly expressed, and yet there's no tumor, because what we've done is co-inject an ion channel that forces these cells into a voltage state that keeps them coupled to the rest of the electrical network, and they simply don't progress to tumorigenesis or metastasis. So we know this is instructive. Now, going beyond the single cell property of conversion to cancer, we could look at morphogenesis, and we can say, okay, here's a tadpole. Let's take a region of the gut. So here, these are some cells in the embryo that are going to become gut, and we're going to inject some ion channels that will set a voltage state that normally is associated with making an eye. And guess what happens? These cells, once you instruct them that way, they will in fact build an eye out of cells that were fated to be gut. And you can make these eyes anywhere, on the tail, spinal cord, in the gut, whatever. They have all the correct layers, the retina, optic nerve, all of that. And also pretty cool is that they instruct their neighbors. So these blue cells here are the ones whose voltage we changed. And they recruited a whole bunch of other cells, which we never touched directly, right? These are just neighboring cells that they recruit to make this nice lens that's sitting out in the tail of this tadpole. And so we can use this technique. We can induce eyes, brains, hearts, and other things. And then there's many things that we don't know how to make yet. So the point is that these bioelectrical gradients are instructive for anatomy. They control at the large scale. And one of the most important features of this is that we don't have to micromanage this process. So what we basically found is an instructive hook into subroutines that say make an eye here, or make a heart here, or make a brain here. We don't have to go in and give all the information about, well, how do you make an eye? What are all the layers? What are all the cell differentiation cascades? What are all the genes that have to be expressed? That would be incredibly difficult. We're nowhere near being able to do that as a field. But what you can do is find the native modular modular control structure in the bioelectric layer that determines which organs get placed in what regions, and then the cells will take it from there. Once you've instructed them what to do, they will turn on all the things they need. So let's look at the planaria, because this is really interesting. In a planarian, you chop off the head and the tail. You got this middle fragment. The middle fragment has to know to build a new head here and a new tail here. How does it know to do that? Well, it has this interesting voltage gradient that says one head here in this depolarized region, one tail here. And if we go in and we artificially manipulate the voltage back here to be depolarized, then sure enough, these cells will build what you said, and they will make a two-headed animal. And these are perfectly viable. So you can make a two-headed animal, you can make a no-headed animal. Okay. So again, instructive. And all we've done is activate the build a head subroutine, and we've done it here at this particular region without actually knowing too much about how you go about building a head in the first place. The cells will turn on and off all the genes that they need once they've received this high-level instruction about what organ they're supposed to build. Not only can you build ectopic heads of the same animal, you can actually build heads appropriate to other species. Okay, so if we take this planarian with a nice triangular head here, chop off the head, perturb the electrical network, and you can get flat heads like a P. felina, you can get round heads like an S. mediterraneum, or you can get your normal triangular heads. And this is, and not only do you get the head shape, but actually the brain shape changes, and the distribution of stem cells becomes just like these other species, because the space of possible outcomes of this electrical circuit contains attractors that correspond to other species that evolution has found in the past. Okay, and you can dial in those without any changes to the genomics. So both in here and in the two-headed animals I showed you, there's nothing wrong with the genome. The genetics are completely wild type. This is modifying the information stored in the electrical network, and you can access other attractors that belong to other species with the exact same genetically normal cells. And in fact, you can access regions of that state space that evolution doesn't use because they're not viable. You can take flat planaria and turn them into these weird spiky forms. You can make these three-dimensional things that look like hats. You can make this kind of thing, which are sort of combinations. So flatworms don't even need to be flat anymore. And so the idea here is that you can really have drastic control over large-scale anatomy given the same genome, because what the genome builds is cellular collectives that use electricity to store their set points, their target morphologies. And this should sound like a very familiar story for neuroscientists, because that's exactly how brains work. So getting to the sort of applied regenerative medicine side of this, unlike salamanders, frogs do not regenerate their legs. And what we've done is we've taken froglets and we've used this information to design a cocktail of drugs that change the electrical state of the wound. And you can see here, normally in the frog, you amputate the leg and there's basically nothing in 45 days. We add the cocktail, the application is actually just for 48 hours, 24 hours, so 1 day. And after that 1 day exposure, it kickstarts the whole cascade. So here, pro-regenerative genes become turned on. So this is MSX1. And then immediately you start growing this leg. By 45 days later, you've got some toes, you've got a toenail, eventually a very nice leg. And you can see that the leg is both touch sensitive and motile. So what you can do is override the normal program of growth, which is normally very little, and give the whole thing a simple signal over 24 hours that then is picked up by all the transcriptional cascades and everything else that will work over months. In fact, our latest paper, 13 months of leg growth following 1 day treatment. Okay, and so we've looked at this so these are frogs and worms. We've looked at this in human mesenchymal stem cells and cardiomyocytes and so on. And it's very highly conserved. It works throughout the tree of life.
A eureka moment.
Gentlemen, we can rebuild him. We have the technology.
Unicorn. Immortality cryptocurrency.
Ouch!
Immortality cryptocurrency. Buy it.